So this is a nice three-dimensional drawing of a biological membrane, and you can already see very familiar components over here. A protein, of course, the cholesterol in the middle, between the phospholipid tails, and the phospholipids themselves, of course, with their heads colored in red and the orange tails. Now there are a few other structures in here which might be a little bit new. You see that some of these proteins have these little extensions branching off here. These are sugar chains, and you can also see the some white lines in here, not part of the membrane, but that's part of the cytoskeleton. So it is not part of the membrane, but it's the cytoskeleton. Skeleton, and the little circles over here, the little extensions, these are sugar chains, and we're going to label the whole thing right now. First of all, proteins that have these sugar chains, like over here, or also this one over here, they have a name, a special name. They're called glycoproteins. Glycoprotein. Let's make the line. So what does glyco mean? The old word for glucose was glycose. And this term is not used anymore, but it's the prefix glyco is still used. And these are basically individual sugar molecules which are linked together into a polysaccharide chain. So you have a sugar chain, carbohydrate, linked to a protein, and then the whole protein is referred to as a glycoprotein. And you see several of these glycoproteins over here. You see over here there's also a sugar chain, but this sugar chain over here is in place of a hydrophilic head of the lipid. Not relevant for us right now, but this is also a carbohydrate chain. So there are several different types of proteins. Some of the proteins you can see are embedded in the phospholipid bilayer, and these proteins which are embedded completely are referred to as intrinsic or integral proteins. They are called integral proteins. Integral proteins. I've seen some textbooks also refer to them as intrinsic proteins, but we stick to the integral proteins for right now. Over here, this protein over here is also embedded in the phospholipid membrane, not all the way through. It only goes through the so-called monolayer, the single layer. But this one is also an integral protein. So I might also label this one over here somewhere like this. Integral protein. And I think it goes without saying that, of course, on the outside over here, this part is hydrophilic. The inside part over here is hydrophobic because it's next to the hydrophobic middle part of the membrane. And this protein over here should also look familiar right now. It's a channel protein specifically. And this one is also an integral protein, of course, because it is embedded in the phospholipid bilayer. Now, if they're integral proteins, then there should also be peripheral proteins. And no, this is not a peripheral protein. This one is also an integral protein. But do you see this one down here? It's on the surface of the membrane, and it does not go into the membrane itself. So this one over here is a peripheral protein. A peripheral protein. It's located on the side. In some drawings, you can also see that the peripheral proteins are connected to other integral proteins. That's also a possibility. So this one also would also be a peripheral protein, even though it does not touch the membrane itself. It touches another protein, but because it's associated with the membrane, you also sometimes refer to this as a peripheral protein. You get the point. So integral protein here, this one over here is also an integral protein because it is embedded. You can actually see it goes all the way through here. So, and there are a few other things I would like to label here. You should label, of course, the cholesterol. You probably already labeled this in a different drawing anyway. You should also label the phospholipid. So that's the how am I going to label this? I don't have enough space. I'm going to draw it around here. This is a single phospholipid. Of course, this over here, the whole membrane is the phospholipid bilayer. Not enough space. Okay, that's the phospholipid bilayer. And cholesterol phospholipid bilayer, you can label the hydrophobic middle part and the hydrophilic heads. I have no space to do this, but I already mentioned this in another video, so please label this as well. And this whole thing here, the whole structure has also a name. It's referred to as the fluid mosaic membrane model. That is the official name of this type of a membrane. There is a second type which was falsified, which was proven wrong, but the fluid mosaic membrane model is now the standard model of a biological membrane. So I'm thinking, is there anything else I need to add over here? Yeah, maybe the idea, did I already mention this, that the membrane undergoes a process called self-assembly. That's a term where I'm going to include this here. So let's put it this way up here, self-assembly. And self-assembly means that if you mix all of these parts together into water and you shake it up, then basically they will arrange on their own into a bilayer. On their own, a membrane assembles itself. It builds up itself without any extra input and energy input from the outside. And this process is referred to as self-assembly. So this was it. No, it was not. I just remember there is a slightly more simplified drawing. Okay, phospholipid bilayer. Now the heading appears. That's the one that I wanted to show you. Okay, that's the simplified version. And over here, this one over here is a glycoprotein. Do I have to write this down again? It's an intrinsic protein. This is a channel protein over here. It's also an intrinsic protein. This protein over here does not go all the way through the membrane, but it's still embedded in the phospholipid bilayer. So believe it or not, this is also an intrinsic protein. And these two over here are not embedded. These are peripheral ones. Peripheral proteins. These are peripheral proteins. And this one over here is a carbohydrate or a sugar chain. A sugar chain. Should I label the whole thing? That's the phospholipid bilayer. The proteins, of course, the individual phospholipids over here. I'm not going to label this. Okay, that's a phospholipid. And what did I forget here? Of course, I forgot the cholesterol, which is embedded in the phospholipid bilayers. I'm going to draw a cholesterol over here, a cholesterol over here, a cholesterol over here, and I'm going to label this with cholesterol. So, and these are intrinsic, just for the sake of completion. Protein. So how are you going to draw that? Over here, I'm going to just make a very quick drawing. You draw a circle and another circle. Please make sure that they touch each other. Another circle over here. A circle here, circle here, circle here. Over here, you have a nice protein. Circle, 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 circle. This one is another protein. Circle. Make sure that the circles are closed. No, I'm not going to call this circle. I'm going to call this hydrophilic head. Hydrophilic head. Hydrophilic head. You get the idea. And hydrophobic tails. That is a way of, no, bad drawing over here. Make sure that the hydrophobic tails, the fatty acid tails, are connected to the phospholipid heads. Okay, you get the idea. Okay, so that is a way of making making a drawing. Make sure that the phospholipids are not too far apart. Some people actually draw them so far apart that there is space. Should not be the case. That is basically already a simplified drawing. Okay, I think if you have a nice drawing of the phospholipid bilayer membrane fully labeled, it's going to probably use about half a page in your notebook. Every time when you make drawings, make sure that the drawing is sufficiently large, because you would like to draw, also have to add labeling and labeling arrows, which are very important. If you do not label the structures, you're not going to get points. That's as simple as that. So take care that your drawing and your labeling is all complete.